participate in CBio conference. My name is Antonio. I work at Sloan Kettering in New York City. And I'll talk about a research that I developed during my postdoctoral work at Columbia University. It's about horizontal gene transfer and how it influenced the evolution of the regulatory code in the microbial world. Because this conference is related with computational intelligence applied to biological problems, and has an audience with wide background, I'll focus on introducing the biological problems. And whenever it's appropriate, I'll mention how you model in a computational framework that would facilitate solving this problem. I start this talk with a quote from Charles Darwin. Although he made one of the most fascinating contributions for biology, in his autobiography he wrote that he deeply regretted that he did not master the principles of mathematics. According to him, people with these skill sets, they have an extra sense to understand reality and the natural world. This is particularly important and relevant for people with a background in computational intelligence or computational biology, because that's what you're going to try to do. We're going to try to translate the biological world with equations that facilitate models, simulate, make predictions, and understand it. The way I like to think is not that one field is more important than the other, but it's the complementation of both fields that expand your possibilities. If you think only in the mathematical terms, you have a set of opportunities and problems that you can explore. If you stay with the biological thinking, you have also a set of problems and opportunities to explore. And if you join both fields, mathematics and biology, it expands the possibilities to novel opportunities that you couldn't think before. Now it's time to go back to the focus of the research and give you background on horizontal gene transfer and later on gene regulation. Horizontal gene transfer is a widespread phenomenon among microorganisms. It's an ability that microorganisms have to acquire DNA from the environment, which increase their capabilities and functional possibilities. It's being proven and observed that about at least around 50% of cultivable ocean microorganisms are confirmed horizontal gene transfer recipients. It happens not only in between prokaryotes, but sometimes in eukaryote organisms. And one mechanism that facilitates the process is endosymbiosis in germlines. In the clinical set, horizontal gene transfer is responsible for spread of drug resistance that makes it relevant for clinical aspect and practical applications. Horizontal gene transfer changes our interpretation of evolution. On the left side of this figure, we have a representation of a tree as thought by Darwin. At the bottom of this plot, there is a common ancestor that diversifies over time, and at the top you see the current organisms. They all could trace back to a common ancestor. However, when you think in terms of horizontal gene transfer, evolution becomes much more complex and convoluted, where a piece of information is exchanged among organisms through all evolutionary process. Horizontal gene transfer can be identified when you analyze the genes that are present in a phylogenetic tree. This figure highlights a schematic representation of a phylogenetic tree, and at the bottom of this tree, it would be the organisms that we see now. And we can sequence their genome and identify some genes. For example, I highlight in red a specific gene of interest. And if you observe, there are 5 instances in the left side of the tree, and one instance that is far away from the tree. And when you observe patterns like this, it's evidence for horizontal gene transfer, because it's much more likely that the gene far away in the tree came from a common from the donor organisms on the left side than if there was created by novel mutations. In terms of computational intelligence, you can model the presence or absence of this gene in each organism with binary terms. So I can write 1 if the gene is present in the organism and 0 if it's not. And you can consider aspects, constraints in the evolutionary tree to infer if the presence of a gene came from a common ancestor or horizontal gene transfer. I'm not going to go in details of these equations, but I can give you a little bit of the intuition. When you have genes that appear in multiple members of one clade in a phylogenetic tree, it's much more likely that they all came from a common ancestor that at some point lost the gene in some organisms but kept it conserved in multiple of them, than if you had multiple instances of de novo mutation or horizontal gene transfer in this cluster. However, if you have a single instance of a gene in a cluster far away in the phylogenetic tree, and this gene resembles much more genes from a distinct group than genes of proximal groups, this makes a strong case that this gene was acquired by horizontal gene transfer and not de novo mutation. That summarizes the background that you need to understand horizontal gene transfer, and now I have to introduce the importance of gene regulation. Inside a cell, the gene must be recognized by the recipient machinery and be transcribed into RNA and translated into protein so that it becomes a functional unity. This was exemplified in a quote in 2000 that the incorporated genes must be expressed in a manner that benefits the recipient organism. The process of recruiting RNA polymerase and activate or not expression, it's what is called gene regulation. I represent schematically the principle of gene regulation in this figure. I illustrate a prokaryotic cell. They contain proteins in the membrane that are able to sense signals from the environment and transmit the signal to regulatory proteins that are called transition factors. These proteins recognize and bind a specific sequence of the DNA, and they are able to recruit the machinery that is responsible for gene expression. This process of regulating when a gene should be expressed or not and the magnitude of the expression, it's called gene regulation, and it's very important for cell adaptation. For example, in environments that a cell are dealing with high dose of antibiotics, they have to express genes that will help them to tolerate or break the antibiotic. But in the absence of antibiotics, it's much better for the survival that they don't spend energy with proteins that facilitate survival under antibiotics condition, and they spend energy in other activities and other metabolisms or growth or division. Moving forward, I can represent schematically more details how transcription initiation occurs inside a cell. The green line represents a strand of DNA, and the shapes on top of it represent a complex unit called RNA polymerase that is responsible for transcription of RNA from the DNA. This protein recognizes a specific sequence in the cell that is highlighted in this figure by this -35 and -10 region, and it has some letters on top of it. The RNA polymerase is able to bind a variety of possible sequences of binding motif. The possible space is diverse, and some sequences are more likely to be bound or not. The pattern that is recognized by the RNA polymerase can be represented schematically by what we call a binding motif, that is a sequence of letters that is recognized by the RNA polymerase. Bigger letters represent patterns that increase the probability of buying highly in small letters patterns that have weak contribution for binding. This can be represented quantitatively, mathematically, with what it's called a position weight matrix, or PWM. I represent at each position a value for the contribution of each letter A, C, T, G, which are the 4 possible letters for DNA, and a probability for each of them. So you have in the first row a probability at position 1 of having each of the possible 4 letters, and that goes through all the sequence until the last character in the binding motif. I provide you the background for horizontal gene transfer and gene regulation in microorganisms. Now it's time to put them together to study the influence of regulatory regions in horizontal gene transfer and vice versa. I represent it schematically in this figure a foreign DNA shown as a dark gray wool ball that is transferred to a recipient organism in the process called horizontal gene transfer, which is highlighted in red by HGT. And inside the cell, this piece of DNA has to be recognized by the transregulatory factors that occurs inside the cell, which are sigma factors or transition factors. These factors, they bind pieces of DNA that are called cis-regulatory elements that are highlighted in red. And they lead the expression of genes to a low level or a high level. If it's very low, the gene is poorly expressed, does not really improve the fitness of the recipient, and it's not selected over time. If it's very high, it's overexpressed, it usually causes a burden to the recipient and it sticks to the cell, and it's lost during evolution. However, there is an adequate level of expression where the gene improves the fitness of the recipient organism. And when that happens, they tend to long-term rotation of the transfer elements and to be observed through evolution. Between the low and high transcription level, the regulatory region can have mutations that may facilitate the cell to achieve the adequate and proper level of gene expression. This is the schematic problem that we are working. We are trying to understand how these regulatory regions actually affect the gene expression of a foreign piece of DNA. At this moment, the idea of gene regulation, the context of horizontal gene transfer, is well defined, and we need to think how we can collect information to understand it better. This joins both computation as well as experimental techniques that I'm going to talk to you without going too much details. However, I'll provide information necessary that you can search in more details in case you're interested. We dig into a public database and we collect about 30,000 genome sequence of regulatory elements from about 180 genomes that would be representative of the microbial diversity that we observed nowadays. These regulatory regions are attached to a neutral protein so that it would not provide different background among them, and we quantify the expression of it at distinct recipients that are phylogenetic distinct bacteria. The quantification is measured by the abundance of RNA that the cell expresses given the amount of DNA that it carries. We use a technique called RNA-seq and DNA-seq. RNA-seq indicates that you count the abundance of RNA, and DNA-seq indicates that you count the abundance of DNA that you observe, and the seq just indicates that this characterization is performed by deep sequencing. We chose 3 phylogenetic distinct organisms with a characteristic that one is independent phylum and 2 of them share the same phylum information so that we can compare within and between phylum difference, and they also have distinct GC content characteristics. The expression level is measured based on the RNA and DNA count. I provide here the equation just to make it more formal. I compute activity in terms of log space. It comes from the ratio of RNA expression divided by the DNA expression. The DNA indicates the abundance of cells that carry that gene, and the RNA indicates the abundance of transcription that you observe. This activity is normalized by the endogenous activity, which is this mu and sigma self, and it just indicates the mean activity of endogenous promoter in the recipient, and sigma self indicates the standard deviation among these endogenous promoters. Endogenous promoters are promoters in our library that come from the recipient organism that we're using for normalization. The first step for analysis was to see how this transcription activity distribution is represented for each organism. The y-axis here represents the density of activity levels, and the y-axis represents different transcription activity bins. What is important in this figure to be highlighted is that if you look at the regulatory regions that is highly transcribed in the 3 organisms, the top 10% of them, and you search for a motif, you observe the same motif pops up in the 3 organisms, which is this pattern, and this pattern is the sigma 70 motif that is the protein important to assemble the RNA polymerase inside this organism. If you look in more details, even if you don't focus in the 10% most active promoters that is shown in red here in this figure, you see sigma 70-like sequence in all possible group of bins. In gray are just sequences that are not statistically significant, so you can just disregard them. And for all recipients that are shown in different rows here, an 80 reach sequence or sigma 70-like sequence is what you observe. The conclusion that we made from this analysis is that the sigma 70 motif is a universal regulatory code shared by all recipient organisms. This provides evidence of a universal code in terms of the binding motif, but the conservation of the regulatory code can be also analyzed at the resolution of the transcription start site, which means the first location the transcription occurs. When you take the RNA-seq that is available in the cell and you sequence them, you can align the sequence back to the promoter that they came from, and the 5' end of the sequence, they are going to represent the transcription start site position. And we can compare this transcription start site among the 3 recipients. There are 3 possible cases. If they all come from the same transcription start location, as shown in the panel on the left side and top, you'd consider them conserved. If only one recipient have a transcription start site distinct than the other 2, we consider them semi-conserved. And the third scenario, where the transcription start site is distinct among the 3 recipients, we consider the transcription non-conserved. And when you make this comparison, we observe that 93% of the transcription start site occurs within the same location in the 3 recipients. Now we have 2 evidence. First, most transcripts occur at exactly the same transcription start site. And the second evidence is that they all recognize the same binding motif, which would be a universal regulatory code shared by all organisms. And we are wondering what is causing that, why that's happening. And we can represent this problem schematically in this figure. The y-axis represents the fitness landscape, which indicates the advantage of a sequence to be incorporated by a recipient organism. At the bottom, it represents high fitness, which is a high benefit for the recipient organism, and at the top, low fitness. And you have two possible spaces. The incompatible space, where the regulatory machinery does not recognize the regulatory regions, there is no expression, and any mutation would be equally non-responsible to any expression. And at the compatible space is when you start to see that the sequence can be recognized by the regulatory machinery and initiate transcription. At the close to the consensus sequence, you'd have the highest level of transcription, and the further you go to the consensus sequence, the weaker would be the expression level. However, in the compatible space, single mutations could drive the sequence to improve the regulatory expression and facilitate the search for an optimal expression level inside the recipient. That's the model that we do, and this is how we simulate. We approximate the regulatory sequence shown on the right side in this figure. Instead of using the 4 letters A, C, T, G and a probabilistic representation of each of them, we approximate them for a binary representation with a sequence of 0 ones that indicates a pattern that could be bound or not. In silico, we simulate distinct regulatory codes that would be proportional to 2 to the number of the bits that represent the regulatory code. In this specific case, we consider only 7 bits, and we'd have 2 to the 7 possible regulatory codes that are represented and the sequence of all 0 until the possibility of all bits being 1. Each possible strain can go to 3 processes. One is birth, when they duplicate itself. The other is a regulatory mutation, where the regulatory code has a mutation and one individual of this population is mutated to a regulatory code 1 homing distance away from the original, and the possibility of horizontal gene transfer. And there is this constraint in horizontal gene transfer that it can only occur among individuals with the same regulatory code. And when we simulate this, we see 2 possibilities. At the top, the y-axis represents the regulatory diversity, and the x-axis would be time in terms of number of generation. When you consider the possibility of horizontal gene transfer, it reduces the overall convergence of the regulatory diversity when compared to a case that does not consider horizontal gene transfer. And that occurs because horizontal gene transfer facilitates the spread of genes that increase the fitness of the recipient organism. This result is important both to understanding fundamental biology as well as for practical implication. In terms of fundamental biology, it shows that the process of horizontal gene transfer constrains the evolution of the regulatory code. And in terms of microbial engineering, one important problem is called biocontainment and safety. You want to be able to engineer strains in the lab, but you don't want them to leak and cause problem and interfere with strains that are in the wild environment. One possibility that you could increase this constraint is designing species with orthogonal regulatory code and regulatory language, and because they wouldn't be able to exchange genes with wild-type species, they would be less likely to survive and less likely to interfere with wild-type species. And I finished this talk thanking everybody for listening, and with acknowledge for the group where I provide this research, which was Harris One group at Columbia University. And I highlight in bold Harris and Nathan, who are the 2 ones that worked directly with me in all this problem. And thank you everybody for listening, and I'm open for questions.